Section 6. Programming a sequence file. Example, CO2 response experiment. To control the CO2, first ensure that columns 1 and 4 are filled with fresh white soda lime chemical. Next, remove the battery compartment cover and, using tool LCM146, remove an empty CO2 cartridge. To ensure that the CO2 cartridge is empty before removing it, use tool LCM146 at the narrow end and press this into the safety valve, which is found next to the electrical connections on the front of the LCpro console. Alternatively, if you know that a filled CO2 cartridge is installed and you wish to swap the cartridge, use tool LCM039 and screw this fully into the valve and then back for one turn. Leave the tool in place in the valve for 30 minutes. This will ensure safety and prevent damage to the rubber O-rings within the cartridge holder. Remove the empty cartridge and fit a fresh one from the 10 supplied in the spares kit. Replace the battery compartment cover securely. From main menu 1, enter Climate, then C-Set. Set CO2 concentration to a high value between 1500 and 2000 ppm. Next, enter Options, then CO2 mode. Set the control mode to Normal. This is so LCpro can accurately gauge the concentration to begin the experiment. We will change this setting later. The source can be set to bottle only for using an installed cartridge. Before placing the leaf in the chamber, you must wait for the desired C-set value to be reached. Monitor the measured parameters screen until C reference reaches 1500. Once the C-set value has been achieved, place a plant leaf in the chamber and leave it to equilibrate at ambient conditions. During leaf settling time, set an initial graph to determine the optimum conditions and timing for the CO2 response experiment. Set the graph to plot A, rate of photosynthesis, and E, transpiration rate, on the y-axis against time on the x-axis. Press Start. The graph will now appear and be updated once A and E reach equilibrium and are in a steady state, i.e. no longer increasing. Reset this graph by using Disable. Select Graph, touch X-axis, and touch to select CI from the drop-down list. Press OK. Select Every as Log. One data point will be plotted upon each record, each step of the sequence. Touch Y-axis on the right and select A from the drop-down list. Press Start. The graph will now appear and be updated in real time. Next, change the CO2 mode from normal to fast. This allows speedy and accurate CO2 concentration changes at each step of the experimental sequence. Next, set a sequence. The sequence function allows an automatic experiment to be set up. Enter main menu 1, touch Sequence. No climate sequence file is loaded will appear. An RGB light unit is fitted. Confirm yes, you want to create a new sequence. Step number 1 can now be edited. Underlined will be dwell. This determines the length of a step. Here we will set the temperature to control at 25 degrees centigrade. Here we can set the total Q or PAR value and the relative contribution of red, green and blue LEDs. We will select CO2 to be 400 parts PM at this step 1. Next, Water vapour, or H2O, we will control at ambient. Ensure that R is selected in options. This will record the data point at step 1. To select a new step, press the downward arrow, and here step 2 is created. Repeat the settings as desired. Each time a new step is created, the previous step's settings are copied in to save time if you wish to maintain values between stages. Select each parameter that you wish to change, in this case CO2 concentration. Use the keypad to change the values and enter. Here we are controlling CO2 at the point of atmospheric CO2 concentration, 400 ppm. We wish to increase the dwell time from 3 to 5 minutes. This is the normal procedure when carrying out an ACI, or photosynthesis rate against intercellular CO2 response curve. Here we have step 8, which will be controlled at 1000 parts per million CO2. We will continue to increase the CO2 concentration through the steps until reaching our final step and returning to atmospheric CO2 concentration. To save the new file, you can name the file using the keypad. Once it is saved, you will be able to scroll through and check all of the steps. Now that the CSET value of 1500 has been reached, we re-enter and start and then run our saved sequence file. We can see the total number of steps and the time remaining on the current step. Now the sequence file has reached step 2, we can see that the first data point from step 1 has been plotted on the graph. Here the value of A is 2.64 and the upper and lower range are given either side of that value. Now we are on to the third step, plotted on the graph, and just moving to step 4 of the sequence file. Now C reference is at 99 and it will come down to 50 for step 5. Nearing the end of our sequence, we can really see the curve now plotted. Here we are on the final step 11 with 8 seconds remaining. As the final data point is recorded, the climate sequence automatically stops. Other settings would be to turn the entire console off at this stage automatically. Additional options on the graphing screen allow you to copy the image as a bitmap, to export the data file associated with the graph, or to pause the graph while you change a setting. Now we wish to turn off the log file so that we can carry on and set up the next experiment without interfering with our saved data. Enter climate, and now we wish to turn off the CO2 control. Return to ambient by pressing amb. We also wish to turn off the light unit, and this can be done by entering 0 in Q total. Now all of the climate controls are set to return to ambient at the end of the experiment. Release the leaf from the chamber and remember to store the chamber open until you use LCpro again.